Hey guys, it's Medicosis Perfect Status, where medicine makes perfect sense. Let's continue our biochemistry playlist. In previous videos, we talked about proteins, enzymes, enzyme kinetics, including competitive and non-competitive inhibition. And we talked about DNA and DNA replication. Today, let's turn our attention to RNA, messenger RNA, ribosomal RNA, and transfer RNA. mRNA is massive. rRNA is redundant, abundant. And tRNA is the tiniest. Look at that, the RNA does not contain T or thymine. Instead, it contains uracil. Please watch the videos in this biochemistry playlist in order for maximum understanding and retention because they follow one another. Your email is sending a message. For this, we need a computer. The hardware is your DNA. And we need a computer code. The software is your genetic code, which depends on the sequence of the nitrogenous bases. AUG is not the same as GUA. What kind of message are we trying to send? Protein synthesis, baby. When you copy the DNA, it's called DNA replication. It happens in the S phase of the cell cycle. When you convert DNA into RNA, it's transcription. And then the RNA will leave the nucleus via the nuclear pores. It will go to the cytoplasm trying to find the ribosome. And then translation from RNA to proteins. After transcription, there is post-transcriptional modification. After translation, also there is post-translational modification. DNA replication happens in the nucleus, and then DNA transcription also happens in the nucleus. The end result is beautiful mRNA. The mRNA will leave the nucleus through the nuclear pores, will go to find those lovely ribosomes on the rough endoplasmic reticulum, and then we'll make proteins. #translation. Thymine is to DNA what uracil is to RNA, which means there is no such thing as thymine on the RNA. Instead, we'll have uracil. DNA replication was discussed before. Remember, in order for me to make another copy of DNA, I need DNA polymerase. I'm always laying down those nucleotides in the 5' to 3' direction. Now let's make some RNA, especially mRNA. Because remember, there are many types of RNA. Messenger RNA is the one that takes the message from the DNA in the nucleus to the ribosome outside the nucleus. And by the way, mRNA is massive. Next is ribosomal RNA. Where do I find it? In the ribosome. Oh, that makes sense. And this rRNA is redundant. It's the most abundant RNA. The third is transfer RNA, which transfers the amino acid into the correct site and then will bind them together by peptide bond. This tRNA is tiny. The only RNA that contains information that is translated into proteins is mRNA. Let's start with my DNA. Here is the original parent coding strand. Let's copy it, #replication. Before you know it, I have DNA template strand. A becomes T because I'm still on the DNA. Next, let's convert the template, not the original coding strand, no, the template DNA strand into mRNA, #transcription. In the first story, the hero was DNA polymerase because I was trying to make DNA nucleotides. But in the second story, the hero is RNA polymerase because I'm trying to make RNA nucleotides. Notice that RNA does not have any T's. Instead, the A will become not T, but U. You're not allowed to say T if you're talking about RNA. Thymine does not exist on RNA. Next, protein synthesis, #translation. Follow the recipe. AUG, this codon equals methionine. GAU, this equals aspartate. You lay this amino acid down, and this one, and you bond them together via peptide bond. Don't forget amino acid. The amino before the acid, the N terminus, is before the C terminus. And then we'll keep doing this until we hit a stop codon. The mnemonics were mentioned in the last video. Please recall that I'm laying down the nucleotides of this RNA in 5' to 3' direction. So the AUG, the first amino acid, is near the 5' end, while the stop codon is close to the 3'. The first type of RNA is mRNA. It's the messenger. It's massive. Why do you call it messenger? Because it carries the genetic information from the DNA in the nucleus to the ribosomes outside the nucleus, mainly on the rough endoplasmic reticulum. How did it leave the nucleus? Nuclear pores. How do I make mRNA from DNA template? RNA Polymerase. In prokaryotes, mRNA is polycistronic. What does that mean? A singular mRNA molecule can give me many proteins. However, in eukaryotes, like humans, mammals, man, monkey, monocistronic. One mRNA molecule yields only one protein, not many. But in prokaryotes, you get plentiful proteins. But in humans, only one. You need to memorize AUG as the start codon, which equals methionine, and you need to memorize the 3 stop codons, which stop the translation. The stop codons are UAA, UGA, and UAG. How about UGG? GG is your mother. Pardon me, I mean it in a good sense. Do you want your mother to stop? No. You want to tell your mother, you go, girl. That's not a stop codon, but UAA is a stop codon. Next, ribosomal RNA. Please recall that the rough endoplasmic reticulum is smooth endoplasmic reticulum with ribosomes. The ribosome is rRNA plus proteins. Functions of the rRNA. Well, it makes ribosomes. Well, no kidding. And the ribosomes will help us develop the rough endoplasmic reticulum, which is the center of protein synthesis, #translation. Moreover, some rRNA are ribozymes. What the flip is that? Zyme means what? Active enzyme. Ribo, because we're talking ribosome. These enzymes are made of RNA, not your regular proteins. I've told you before in my biology playlist that all your channels are proteins, all your pumps are proteins, all your receptors are proteins, all your carriers are proteins, and all your enzymes are proteins. But I lied to you because there are exceptions. Some enzymes are not proteins. Some enzymes are just RNA molecules, and these are called ribozymes. Next, this rRNA will help catalyze peptide bond formation between one amino acid and the next amino acid, and it splices out its own introns. Remember, what's the difference between introns and exons? Which ones are the good guys and which ones are the bad guys? Well, exons are expressed. They are the good guys. But introns interfere with the process. So these are the bad guys. And when you splice out your own sins, that is order. Otherwise, you descend into chaos, you bloody monster. Hey, Medicosis, where was the rRNA made? Place of birth is in the nucleolus, which is in the nucleus. Third, tRNA, transfer RNA. What are you transferring? I am transferring the message from the codon on the mRNA into the amino acid because I will select the amino acid of choice and insert it here. The codon is on the mRNA, but the anti-codon is on the tRNA. And the ribosome with its large subunit and small subunit is hugging both of them. Just like your DNA, phosphate is on the 5' end. The 3' end has hydroxyl group. And on tRNA, the 3' always has CCA. Let's activate, i.e. charge, our tRNA. What do you mean by charge it? I mean load it with an amino acid, okay? Who's gonna do this? A beautiful enzyme known as aminoacyl tRNA synthetase. I love it. It's gonna load the amino acid onto the 3' end of the tRNA. Each amino acid has its own aminoacyl tRNA synthetase enzyme. Anytime you say the word synthetase in biochemistry, synthetase requires ATP. And ATP will enter into the reaction. Oh yeah, and we'll convert it to ADP. Worse, we will milk it dry and convert it to AMP. Oh, What's the difference between tri and mono? 2 phosphate groups, 2 inorganic phosphates left the chat, which means 2 high-yield energy bonds were utilized to release energy enough for us to attach the amino acid to the 3' end of the tRNA, making a lovely energy-rich bond. The energy here is going to be used to make a peptide bond between this amino acid and the subsequent amino acid. Mnemonic time. tRNA gets activated, i.e. charged, when amino acid is attached to its 3' end, thanks to the amino acyl tRNA synthetase, which requires ATP, namely 2 high energy bonds. You load the truck with cargo at the rear end of the truck, but you load my tRNA with amino acids at the 3' end of the molecule, just next to CCA. Pause and review. What are the characteristics of the genetic code? Unidirectional, universal, unambiguous, non-overlapping, and degenerate. I hope by now it's clear that we go only in one direction, from 5' to 3'. Next, universal. What does that mean? It means it's universal across species, which means AUG always codes for methionine, whether we're talking about AUG in a dog, a cat, monkey, or man. Doesn't matter. It always equals methionine, and that's universal. Next, unambiguous. What does that mean? It means that one codon will give you only one amino acid. For example, AUG can only become methionine. AUG can never become anything else. It's unambiguously methionine breeding. Also, I want you to make the link between unambiguous and monocistronic. What does unambiguous mean? It means one codon always gives one particular amino acid. What does monocistronic means? It means one mRNA molecule yields only one protein and not more. Next, your genetic code is non-overlapping. It's commaless. You go in order: A, U, G, G, A, U, etc., etc., etc. Hey, can I skip one? No, we cannot, because we are commaless, we're non-overlapping. Can I read this A once and then read this U twice or thrice? No, because we're non-overlapping. Next, the genetic code is degenerate or redundant, which means the same amino acid could be coded for by multiple codons. Let me explain. How many nitrogenous bases do we have? We have 4 nitrogenous bases. We have adenine, we have uracil, we have cytosine, and guanine. Only 4. And each codon has how many positions or how many slots? Well, only 3. Only 3 bases are allowed per codon. So you go for power 3. 4 types of nitrogenous bases to the 3rd power, because we only have 3 spots in a codon. 4 power 3 equals 64. Oh, so we have 64 codons that work and give amino acids. Shut up, you forgot the 3 stop codons, UAA, UGA, UAG, which means 64 minus 3 stop codons, which do not code for any amino acids, equals 61. And how many amino acids does your body make? Only 20. So we have 61 codons, but only 20 amino acids, which means some amino acids will be coded for by multiple codons. Let me ask a question for the math nuts out there. Assume that your credit card has 16 digits, and we only use these numbers in English, or Arabic numerals to be more specific, which were discovered in India to be historically accurate. The question is, how many possibilities, how many rearrangements do banks have? Can you let me know the answer in the comment section? Next, the genetic code and the wobble. The wobble is nothing new. The wobble belongs to degenerate. Whenever you see wobble on your exam, just remember number 3. They are referring to the 3rd position. Because look, the third position here is variable. The first and second were constant, and the end result was always alanine. This is actually very beneficial for you. Suppose that you made a mistake while putting those nucleotides, and the mistake happened in the third position. Instead of U, you put C. Well, you're still going to have alanine. Or you put A, you're still going to have alanine, which means at the end of the day, you will not feel it. You will not have any disease, i.e. it's a silent mutation. It's a degenerate mutation. Mnemonic time. For wobble, just remember the triple Whopper at Burger King. Because if you go to Burger King, if you order anything, they will get your order wrong. But if you order the Whopper, they are very good at making the Whopper. You will not feel any mistakes. It's a silent mutation. Do you want a crazy mnemonic about the fact that your genetic code is degenerate to help you remember it on the exam? It is a horrible mnemonic. Forgive me, I'm just trying to help you remember it. Suppose we're talking about a degenerate person, i.e. promiscuous, meaning sleeps around like crazy. Well, we have no idea who the daddy is. A single baby can come from many daddies. We have no idea who's your daddy. Isn't that degenerate? And wasn't that behavior redundant throughout history? Next, mutations. To get the most out of the next section, please watch my video titled Mutations in my biology playlist first. In that video, we discussed those definitions which are important for your exam. And I've told you that a mutation is a change in your DNA sequence, i.e. the computer code is toast. Instead of sending the correct email to your friend, it might send the wrong email or it might send nothing. The mistake could be irrelevant. It could be a minor mistake, or it could lead to not sending any emails. Or worse, it can lead to deterioration in the relationship between you and your friend down the road from now on and until the end of times. Let's take a step back. Mutations could be nucleotide mutations, which is today's topic, or chromosomal mutations, not today's topic. Go watch my biology video. The nucleotide mutations could be silence mutations, missense mutations, nonsense mutations, insertion mutations, or deletion mutations. The first three are collectively known as point mutation. The last two are frameshift mutations. The first three will affect you in one nitrogenous base, in one nucleotide, in one codon. It is pointed. But the frameshift mutation will affect you now and then until the end of times, all the way down the road. Let's start by the nonsense. Anytime I hit a stop codon before it was due, i.e. a mutation led to a premature stop codon, I'm gonna stop the translation, and the mnemonic is stop the nonsense. Next, point mutations also include silence and missense mutation. How about silence mutation? Well, you changed one nucleotide in one codon. In the good old days, I used to get leucine. Now I got also leucine. Oh, you didn't feel anything. It was a silent mutation. Amazing. That's the beauty of the wobble, the triple whopper. Hey, medicosis, how come the first and second position are that unique and they are not affected, and the problem usually happens in the third? That's a long story, my friend. I'll just give you a hint. Usually, in the first two nucleotides, we require specific base pairing. Next, missense mutation. Just remember, missense, misplaced. That's the mnemonic. Missense, misplaced. An amino acid got misplaced. I should have put glutamic acid here. Instead, I put valine. Oops, I misplaced the amino acid. Missense mutation. This is seen in sickle cell disease, a very common exam question. So sickle cell disease is a misplaced amino acid at one position only. It's a missense point mutation, namely a nucleotide mutation. Why do you call it point? One nucleotide got affected only in one codon, in one position. Now let's zoom in to talk about the frameshift mutation. Here is an example of frameshift deletion. Normally it should go ATG, TTA, CCTA. Alright? Amazing. The problem here that this second T got deleted from my DNA. Oops! This was supposed to become mRNA, which was supposed to code for leucine. But now look, what's gonna happen? I'm gonna borrow the next one. I'm gonna go T. This one got deleted, so TAC. TAC codes for tyrosine. Not only that, the next one is disrupted too. CCT used to be proline, but because of the shift in the frame shift, it's gonna read leucine, and everything down the road can end up causing another amino acid to be here. That's why frame shift mutations are worse than point mutations for the most part. Remember, each one of the amino acids has a 3-letter abbreviation and a 1-letter abbreviation. At the very least, you should try to memorize the 3-letter abbreviations to all of them. In sickle cell disease, at a position, I was supposed to put glutamic acid. Instead, I put valine. Oopsie. This usually happens at position 5. Instead of glutamic acid, I put valine. The end result is sickle cell. This is a missense point mutation. Missense, misplaced. Is this conservative? No, I did not conserve the amino acid. Glutamic acid became valine. This is non-conservative mutation. Why do we call these point mutations? Because it only affected one nucleotide in one codon, which resulted in a different one amino acid. Mnemonics. Missense is misplaced. Mnemonic. Hemoglobin SS, which is sickle cell disease, is a missense mutation. But hemoglobin AS, if you remember, is sickle cell trait, not sickle cell disease. Which one is worse? Of course, hemoglobin SS, the sickle cell anemia, is way worse than the sickle cell trait. Another mnemonic. Look at those sickle cell. They are sharp and pointed. Point mutation. If you want to learn more about sickle cells, I have many videos about this topic in my hematology playlist. Pause and review. Quiz time. Can you answer this? Let me know in the comment section. You will find the correct answer in the next video in this biochemistry playlist. If you like this video, you will enjoy my general pharmacology course where we discuss pharmacokinetics, pharmacodynamics with all the graphs and the equations. You can watch the 60 minutes of the course for free on my website, medicosisperfectionalis.com. To learn more about how your kidney works, download my renal physiology course at medicosisperfectionalis.com. Learning is fun. Remember that medicine makes so much sense once you understand what the flip you're talking about. Thank you for watching. Please subscribe, hit the bell, and click on the join button. You can support me here or here. Go to my website to download my courses. Be safe, stay happy, study hard. This is Medicosis Perfectionalis, where medicine makes perfect sense.